So Curtis is one of the most influential figures in computational microbiome science. He's a professor of computational biology and bioinformatics in the Department of Biostatistics at the Harvard Chan School, with additional appointments in immunology and infectious diseases. He co-directs the Harvard Chan Microbiome and Public Health Center, leads the NIH HCMPH Centers for Characterizing the Gut Microbial Ecosystem in Inflammatory Bowel Disease, and was a core architect of the original Human Microbiome Project. His group has driven many of the foundational tools in multi-omic analysis, including Human2, Metaflan, and the broader biobakery ecosystem that is now one of the standards in the field. His work spans functional metagenomics, microbial community modeling, host microbe interactions, structured machine learning, and many other things. I'm very happy that he's here today, and I look forward for our continuing collaboration between Penn State and Harvard. And we have a little token of appreciation, a little plaque. Please welcome him.
This is amazing. Thank you so much. It's not just the socks. I do have the Microbiome Center socks, which Erica was kind enough to bring in Boston.
You have 2 pairs of socks. We've learned, yes.
And as I was saying last night, my family's from Western Pennsylvania. It's been very nice to be back to see familiar landmarks. I took my driver's test near here. So yeah, we got to drive by the state prison, which I remember.
I remember the outfit.
It's good to be back. It's really nice to see so many. I wanted to start with a poll, since I was also delighted to participate in the Microbiome Day webinar a few months ago now. How many people overlap with that talk, if anyone? Okay, the front half. I used the same title, but I've been telling everyone I did switch out all the contents. So same projects, different stories. Anyone who saw that talk, it'll be mostly different this time.
Good to hear it twice.
Yeah. So what I'll focus on today that's a little different than at the previous version is what the field has come to call microbial or microbiome dark matter. The phrase that you'll see used in a lot of these very evocative primary literature titles. We had a really nice meeting on microbial dark matter a few months ago now at Irvine with the Zymo company. It's entered the popular press. So when you see popular science articles about microbial communities, you'll see how much dark matter there is, how much we don't know. And when I was making this slide a few weeks ago, I found out we now have a whole book on microbial community dark matter, which I will have to read. I was not aware of this. And there are many ways to define this, but for most of this talk, I'll define it as uncharacterized molecular function in microbial communities, mostly protein function. So things like enzymatic substrates and what proteins are doing in terms of structural pathways or regulatory pathways. And this is not a new problem. So you can go back several decades now to when we first started sequencing anything, let alone started sequencing lots of bugs in microbial communities, and find great quotes like this saying like, hey, just because we can sequence something doesn't mean we know what it's doing. So fast forward from yeast and E. coli decades ago till now, and most of what we sequence in microbial communities, we don't know what it's doing. And that's becoming a problem as the microbiome field broadly becomes more applied, becomes more translatable. Over the past few years, places like the FDA have newly developed regulations around things like live biotherapeutic products, saying, well, actually, we realize we can't figure out mechanism of action in most cases yet, but hey, that should be a priority. So if we want to understand how some of the recently translated microbiome products actually work, or if we want to start rationally designing or identifying things like new small molecule therapies based on microbial chemistry, these are all cases in which understanding the molecular function of everything as much as possible in the human microbiome, in other microbial communities really matters. So I'll highlight a couple of different dark matter areas that we've been working on today, one of which is the Human Microbiome Program. And it's getting towards the end of the year. I need to update this slide now that the government is open again. As of January, so almost a year ago now, this was a newly launched program from the NIH Common Fund with the mandate to characterize the baseline human virome, much as the Human Microbiome Project characterized the baseline human microbiome. I am delighted to be running the coordinating center along with Owen White. So we get to do fun things like make sure the community has access to the data and resources from the project. We get to do boring things like organize all the meetings. But the consortium consists of 5 large centers, each of which has access to different large human populations and is tasked with surveying the body-wide human virome in some definition of health or baseline conditions, so sort of outside disease-related perturbations. There are then another 5 really targeted biological functional studies and another 5 methodological or method development projects that aim to deep dive different areas of virome biology or build better computational or experimental tools for studying the viral components of microbial communities. So putting all of this together, we're just starting to get to the point where some of the large centers are ramping up data generation. Some of the smaller projects will take a little while to build new tools, say, that can be used as parts of these studies or by the broader community. And our goal at the coordinating center is to make sure that those resources are available to the outside world. So I talked a little bit at my previous talk about some of the bioinformatics methods development that we're doing as part of the coordinating center, including better ways to see viruses in plain vanilla metagenomes and metatranscriptomes in the first place. So we've just recently finished a tool called Baklava that, like most of our tools, has a complicated acronym. And I talked a bit about how it worked last time. I'll focus on more applications this time. But to give a very brief overview, Baklava, unlike, say, virus-like particle VLP enrichment methods, which typically perform viral discovery by experimental enrichment followed by assembly to find new viral genomes, Baklava focuses on finding, with very sensitive detection, whatever viruses we can in normal metagenomes or metatranscriptomes. We combine 3 components: a nucleotide-based component that builds on marker sequences similar to what we do in Metaflan for the rest of microbial community profiling. So for each viral species that we're targeting, we build nucleotide sequences that we have reason to believe are unique to that virus. So those are very precise, but maybe not as sensitive as we'd like, since viruses change very quickly, evolve rapidly, have a lot more sequence diversity than bacteria typically. So we have a protein component that again sort of builds on technology from our Human functional profiler. In Human, we're typically looking for proteins in microbial communities, again, to figure out what they're doing and put pathways back together. In Baklava, we use a similar approach, but instead to find proteins that are typical of different viruses, but which we can detect a little more sensitively given amino acid homology rather than just nucleotide homology. And then we put these together with a third component that's based on assembly and deep learning to classify really novel viral sequences. So if we can put together a long enough sequence and say, this is probably viral and probably from this general area of taxonomy, that's an even more sensitive, less specific way of finding viral sequences. So at the end, we put these 3 together to get an integrated virome profile from metagenomes and metatranscriptomes, much like normal taxonomic profiling would provide for the rest of the community. So I talked a little bit last time about how that algorithm works, about some of our applications in inflammatory bowel diseases. We've been starting to look at viral profiles in several different kind of chronic health human population conditions. So another one has been colorectal cancer, building on work that Nicholas Sagata's lab did, and we've followed up in collaboration, putting together as much colorectal cancer shotgun metagenomes as we can. So this now consists of about 15 studies. It's a little more, so it must be up around 18 now. It's a little more than is in this slide. We've hit between 3,500 and 4,000 total shotgun metagenomes. They span a wide variety of different countries, protocols, populations, stages and degrees of colorectal cancer, all the way from controls through adenomas and late-stage colorectal cancer, And then a wide range of different kind of environmental and biometric and exposure phenotypes as well. So we have everything from unfortunately very early onset CRC through adult and later onset CRC. So Nicola's done a lot of great work on building microbiome biomarkers for colorectal cancer. We've done some work on the specific, again, kind of mechanistic strain level phylogenetic components of this in the non-viral microbiome. We can now apply baklava to the same data set, the same populations, and really start seeing different types of ecology that manifest in the virome associated with these microbial communities. So again, I didn't go into some of the algorithms, but we quantify viruses in units of viral genome bins, or VGBs, which are harmonized species-like units that cluster together similar enough viral genomes from a variety of different sources. So if you see VGB, think of it as more or less a viral species. We see thousands of unique viral species-level features across these few thousand metagenomes, most of which are novel. And this is where we start tying together sort of different types of microbiome dark matter. As we've now been able to better see viruses more sensitively across more different conditions, the vast majority are phylogenetically and thus taxonomically novel. We'll see in a few slides, they're also more inter-individually variable, such that out of several hundred unique viruses per sample, many of them are particularly unique. So out of these thousands of virome features, only a few hundred reach even a very minimal prevalence level across this very diverse kind of global CRC population. And the vast majority are phage. So as we look in human gut microbiomes, at least, this is actually even a little low. I'd say 90+% of most populations' viromes are phage associated with typical human gut bacteria. But we do see some eukaryotic viruses, like previous studies, we'll see especially RNA-based plant pathogens that kind of make their way through in association with dietary elements. And we'll occasionally see some viruses that infect other microeukaryotes like Saccharomyces or Candida in the gut. And you can see over on the right here that if we take these viral features, we can do things like look at their ordinations or ecology or population epidemiology, much as we would with typical taxonomic profiles. So there's no global shifts associated with colorectal cancer. You wouldn't expect that. It's the same as with the rest of the microbiome. We can find very strong CRC-associated bacterial biomarkers, but CRC, even in later stages, is not the main driver of overall gut microbiome ecology. And the same thing manifests in terms of the mostly phage that we see. To give you a sense of what sort of a slice off the top of these data look like, some of the ecology of mostly phage in all of the data sets we've looked at does closely mirror their bacterial hosts. So much as you'll see some common bacteria that vary gradually across the population, you'll see phage of mostly these same common bacteria. Many of these are crass phage that are typically Bacteroides phage. Some of them are E. coli, lambda phage, or occasionally Firmicutes phage. Most of them are unknown. And actually, since we made this slide, we now strongly suspect that these top two that are very prevalent are actually phage-like plasmids instead of proper phage. And we're trying to tease those out of databases. Many of the recently published large-scale viral assemblies seem to scoop up these phage-like plasmids as well. So we can look at this at sort of a global population level. In this particular study, though, what we're more interested in are which changes in the virome associate with colorectal cancer development. So much as with other population studies, other types of microbiome profiles, we built statistical tools that will associate microbiome features, taxonomic features, functional features, with different covariates, exposures, or in this case, health outcomes like colorectal cancer. So this is what the output of our current version of our Maslin tool looks like, which is our statistical framework for these tests. I've highlighted down here at the bottom several of the viral features that are specifically CRC associated, typically depleted. So as you see either purple or blue, abundance changes or presence-absence prevalence changes. If they move to the left here, that means those features tend to be less abundant in purple or lost in cyan during colorectal cancer. This is a stronger signal than we'd see for purely bacterial loss during CRC. We see more enrichments, actually. We see a very similar signal in IBD, where IBD was one of the first GI chronic conditions where dysbiosis in the gut microbiome consists mostly of depletion of a variety of different taxa. And there we see all of their phage go along for the ride. They are depleted at the same time. Here, the virome dynamics look to be doing something a little bit different than the typical CRC-associated bacteria. But as you look down the left column here, you see unknown, 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 unknown again. You know, most of these are phage, but they're not phage that have been previously isolated, taxonomically described, and often they're phylogenetically fairly distant from the most closely related relatives. But not always. So if you have good eyes at the bottom there, you might have seen a Totatus virus on the previous slide. This is one of the more negatively CRC-associated phage, and this is a Faecalibacterium prausnitzii phage. F. praus is one of the more typical health-associated, kind of strictly anaerobic microbes. Not itself typically depleted in CRC. It tends to go away in more inflammatory conditions like IBD. But here we're seeing either copies of the associated phage or strains that tend to carry the prophage are specifically depleted during CRC. And we can't differentiate between those two things. We intentionally don't distinguish between integrated prophage and free viral particles because you can't using just sequencing. On the other hand, there are some of these uncharacterized VGBs that are not obviously phage of the usual suspects associated with CRC, Fusobacterium nucleatum, Hungetella, some streptococci, but they are phage that again seem to be either more free viral particles or more integrated prophage in especially later stage CRC in this example. So we're seeing kind of some ecology in the virome that parallels that of the host bacteria, since most of this is phage. We're seeing some that's then also distinct, either because of lineage-specific prophage integration or because of changes in prophage activation and free viral particles. And we see similar stories in IBD. One of the other things I've been emphasizing, though, is that as we've started looking at this and I've been presenting some of this material, people will look at all of these unknowns and say, how is this useful? What can we do with this if we detect a bunch of uncharacterized viral features? And that gets back to a different type of dark matter that we've been dealing with for a variety of circumstances, is saying, well, if we see all of these features, if we see all of these proteins and we don't yet know what they're doing, how can we start to get a handle on that computationally without having to do isolation or genetics or experiments on every single one of millions of proteins? So one trick that works for viruses that leads into some of our other work in the area is to take the set of annotated viral proteins, as to some degree we can annotate all of these hundreds of thousands of viral genomes, get a sense of which proteins they're carrying, and in a subset of cases, we will know what those are. They'll be things like capsid proteins, tail proteins, or in this example, anti-phage defense systems, because many phage carry toxin, antitoxin, or similar systems to prevent other phage, competing phage, from infecting their hosts. So in the case of colorectal cancer or other phenotypes of interest, even if we don't know what these viruses are, we can ask, are they enriched for subsets of viral proteins that do have known functions? So in the case of something like anti-phage defense systems, we do get both significant enrichments and depletions of different subsets of phage defense systems. So in this case, there are phage that tend to be more abundant during colorectal cancer, that tend to carry specific anti-phage defense systems to then kick out other phage during CRC. We don't yet know the ecological reason, but just because we don't have names for these viruses doesn't mean we can't make some educated guesses about what they're doing. So whatever the subset of mostly phage that are CRC enriched are, they tend to not like certain other phage. And we can tell that even without knowing their taxonomy. This transitions into other work we've been doing to better characterize similar like protein functional dark matter, building mostly on another different acronym, a project that we finished a few years ago called the Human Microbiome Bioactives Resource, or HMBR. So the HMBR kind of precedes any of our Virome work and was really built to address this question of protein functional dark matter broadly throughout microbial communities. We have a review from an embarrassing number of years ago now that put some numbers on, even ignoring viruses, how many proteins or what fraction of proteins in a typical microbial community can you assign some biochemical function to versus nothing. And back at the time, using fairly generous definitions of biochemical function, so can we assign any enzyme classification to a protein? Can we assign any gene ontology pathway to a protein? Back at the time, we got about 1/2 to 2/3 of typical human gut communities. So this is a very well-studied ecosystem. Even here, about 1/2 to 2/3 of proteins in each microbial community remain unannotated, uncharacterized. A few years after that, we followed up with a broader set of microbiomes where if we reprofile the same Human Microbiome Project, very well characterized healthy human guts, we get a similar fraction of about 50% to 2/3 of proteins are uncharacterized or completely normal. But the problem just gets worse the further outside of the human gut you go. So yellow here is either completely lacking significant homology to anything characterized or possessing homology to only uncharacterized genes. And by the time you get out into these very tricky environmental communities, the plot is mostly yellow. So we do want to address this. One of the projects that Kelsey in the lab just finished up recently was trying to put some bounds, some modern quantification on this, some bounds of what can we easily know from sequence level features and homology in a range of well to less well characterized environments, and how far towards saturating these environments have we gotten. Part of this was inspired by the degree of functional uncharacterization. Part of it was inspired by sort of the flip side, the glass half full of the field has gotten much better at cataloging and annotating both taxa and functional features in the past several years. So just inside of our own internal databases that we use for building our tools and cataloging species level genome bins, so bacterial taxonomic features, these have gone up by about an order of magnitude in the past few years. That comes almost entirely from metagenomic assemblies. So USGBs or unknown SGBs only have sequence representation from environmental samples. KSGBs are coming from isolate microbes, and those have kind of flatlined in terms of new phylogenetic diversity that's being sampled. That has allowed us to pretty much saturate coverage, at least phylogenetic and taxonomic coverage of these well-characterized environments like the human gut. So over the past few years, we've gone from say 2/3% purely phylogenetic coverage, so not functional characterization, but phylogenetic coverage, to almost 100%, even of kind of very globally diverse human gut populations. So that's good. We're definitely making progress there. There are other areas like companion animals where things started out a little grim a few years ago. So typical reference databases had very modest coverage of cats and dogs and livestock. Thanks to some focused effort in the past few years, that has also gotten much better. But we started from not such a great place very recently. And most other environments that were yellow on the previous slide have improved by several fold, but they were starting from such a low point here in yellow that several fold means that they're only 95% unmapped rather than 99% unmapped at this point. So we can think about this as, again, kind of a glass half empty, where difficult environments, microbial communities, are not just functionally uncharacterized, but really phylogenetically and taxonomically uncharacterized still too. There's multiple different kinds of dark matter there. The glass half full of this, though, is that some of these other environments have very rapidly progressed thanks to information gleaned from bulk metagenomic sequencing. So to the degree that we can continue to target more diverse environments and take advantage of metagenome assemblies from those environments to a sort of approximation, once you assemble a bug someplace, you never need to assemble it again. You can take advantage of that assembly to find the bug and characterize the bug in other environments. So if you have your favorite rare organism, if it's abundant in just one community out of 100,000, the tools now exist for that to be sufficient for a field to assemble that bug out, annotate and take advantage of those assembled sequences, and figure out things about that bug in other environments. So it is possible to advance these difficult environments very quickly. And just kind of as an aside, this is thankfully not just our problem. It's kind of an every tool problem, or even just an every reference sequence problem. We're really lacking assemblies and references for these difficult environments. A lot of the examples on the previous slide came from just our own tools and databases. We get similar results when we compare just about every other reference-based or assembly-based method for profiling microbial communities. So I won't go into the details. There's kind of a spectrum here from purely reference-based tools through purely assembly-based methods, through taxonomic-based methods like K-mer comparisons or sketch-based comparisons. When you apply any of these to well-characterized environments like the human gut, they tend to agree very closely. So we need to do comparisons like this since we don't have thousands and thousands of gold standards where we know the right answer, but we do have thousands and thousands of communities where we can assess agreement between different methods. So here, for example, we're coloring by profiling methodology. The lines show a couple of examples of the same community profiled by several different tools. And in these kinds of environments, the lines tend to be short. There's agreement between different tools for any given community. So again, that's good. This tends to work well in well-characterized environments. And again, things get a little dicey when you go out into companion animals that are somewhat less well-characterized, and really dicey when you go out into challenging soil environmental communities. Long lines, very wobbly lines, and lots of separation by color. So the different methods are more like themselves than they are like the biology in this case. So currently we know, just as we saw on the previous slide, we have kind of a ways to go on these less well-characterized environments. There's more different types of dark matter there. We can quantify it in a couple of different ways. So we've moved some of the unknown unknowns into known unknowns at this point. And the glass half full is that as we start to incorporate more metagenome assemblies, we can move the needle on this fairly. The other type of dark matter that the HMBR in particular was looking at is more protein functional dark matter again. So rather than this side story on taxonomic dark matter. The main goal of the HMBR, which is a collaboration among several different groups, was to broadly identify bioactive components of the human gut, where bioactivity was defined as either causality for a phenotype, a human host phenotype, or mechanistic association, so responding to a phenotype, for example. And that could be either in whole microbes that were, say, pro-inflammatory or anti-inflammatory when present. It could be microbial proteins, which I'll focus on for the next few slides, or we had some work on microbial chemistry as well, and identifying enzyme function and different chemical transformations that produced or abrogated bioactivity in the underlying small molecule. So focusing on protein function and protein level dark matter, Yensong in the group did some work that I'll go very briefly that's published a few years ago. First of all, to even just figure out what the important uncharacterized proteins were, which ones were likely to be bioactive in the first place, even if we didn't yet know what they did. This particular method, again, complicated acronym, was called MetaVibla. And again, not to go into too much depth, since this is published a while ago, but MetaVibla integrates several different data types along with sort of epidemiology and phenotypic information either from humans or from other environments, to make an educated guess as to which proteins, even if they are not yet functionally characterized, might be causally or responsively involved with a phenotype. So it prioritizes out potentially bioactive proteins. I bring this up to give a sense of just how much dark matter there still is of this type, even in the human gut. If we look at the 1,600 or so shotgun metagenomes from, in this case, the second wave of Human Microbiome Project data, these are drawn longitudinally from a set of IBD patients. And the phenotype won't really matter for our purposes today. You can keep in mind that these are about 1,600 metagenomes longitudinally from about 130 individuals from several different inflammatory phenotypes. From these 1,600 or so metagenomes, we assemble out at least partial contigs covering around 45 million complete corpus, so complete open reading frames. As a side comment for anyone who's looked at things like gene cataloging in microbial communities, incomplete fragmentary open reading frames cause lots of problems. Don't use them. But if we look at just the complete open reading frames on any assembled contig, not just kind of complete mags, complete genomes, we get about 45 million unique sequences that cluster down to about 2 million nucleotide level gene families, so at a fairly high level of nucleotide homology, which further cluster down to about 1.5, 1.6 million protein level protein families, amino acid level protein families based on near full length amino acid homology. So focusing on these 1.6 million or so protein families that are typical of this population, this human gut microbiome population, we still get a similar level of lack of functional characterization as we showed several years ago. 500,000 or so of these protein families have fairly high homology to anything in UniProt that has any kind of functional annotation. Again, being very generous, enzyme classification, gene ontology, annotation, anything. Interestingly, another 400,000 or so have very high homology to something in UniProt, but it's something that is still uncharacterized even in whatever isolate microbe it happened to come from, for example. Another 500,000 or so have homology to anything in UniProt, but not high enough to make a good guess as to their function based just on that sequence homology. And even in this very well-studied population, another 200,000 or so proteins don't have significant homology to anything in UniProt, let alone something that's been characterized. So again, this ends up being about 2/3 of the protein families that we see here don't yet have functional annotations. So we know from MetaVibula that a subset of a couple hundred to a few thousand of these are bioactively implicated in inflammatory bowel disease, the phenotype here. But unfortunately, just because we have reason to think a protein is phenotypically important or bioactive, MetaVibula doesn't yet tell us what it might be doing. If we can't pull information out of a catalog or prior experiment or sequence homology, that's kind of where we left off with this previous phase of the project. Yansong is amazing, started her own position in China recently. She went on to say, well, what can we do computationally rather than doing 1.6 million new experiments? What can we do computationally to take advantage of microbial community data and make educated guesses about some of these proteins' functions? And this is building on work that goes back many decades now, using similar ideas in isolate organisms. Again, going back to some of the earliest sequencing in the 90s and early 2000s to say, if we just sequence yeast and we want to know what 5,000 of its proteins do, can we take advantage of a bunch of different genetic data, a bunch of different gene expression data, a bunch of different protein interaction data, and make better informed predictions? And Manuel will recognize this from 20 years ago in our work at Princeton. So we're finally getting out of yeast into microbial communities. So the follow-up project that Yansong put together is called FGOSM, complicated acronym. What it does in practice is exactly what we just described for isolates, but for every microbe in whole microbial communities, and leveraging not just isolate information, isolate functional experiments, but whole community functional information, especially leaning pretty heavily on metatranscriptomics. So now that we can get RNA-seq for whole microbial communities, the protocols are still tricky, so there's not a lot of data out there. But now that we can get that, very much like RNA-seq gene expression for individual microbes that is very functionally informative, we can combine that with other types of similarly high throughput functional information for whole communities, things like co-adjacency. So do genes tend to co-occur with each other in operons? Do they tend to co-evolve? Are they gained and lost at the same time? Do they tend to have similar secondary sequence structures, protein domains? Do they have predicted domain-domain interactions? And pile on different layers of evidence that can be combined to predict potential protein function. And the way that Fugassem does that is, again, analogous to what we did years ago in yeast and E. coli. Fugassem will turn each of these data types into different kinds of interaction networks. So how to do this is pretty obvious if you're working with protein-protein interaction data, but for something like metatranscriptomics, we turn that into a co-expression network. For something like sequence similarity, we turn it into a sequence similarity network, and so on and so on. And then Fugassem uses 2 layers of machine learning to then combine these networks. The first layer learns how much to trust different measurements within each data type. So if 2 protein families co-express with each other at a level of 0.9 versus 0.6 versus 0.3, how much can you trust those different values? If 2 protein families are sequence similar at 90% versus 50% versus 30% homology, how much can you trust those different measurements? So the first layer of machine learning kind of calibrates individual data types measurements. The second layer then combines different data types. How globally functionally informative is co-expression? How globally functionally informative is protein homology, and so on and so on. That gives us integrated networks that we can then use to predict membership function in the sense of pathway membership. For all of these uncharacterized proteins, which pathways can we potentially assign them to with some confidence? And this ended up working better than we expected. In some cases, actually better than it does for isolates. To go into some of the computing for this example, we're looking here at cross-validation across a small subset of known experimentally validated protein functions, spanning both different versions of Fugassem, incorporating more or less data, comparisons with string, which is a database that does something very similar for isolates, using just isolate-based functional information, and then 2 of the most recent best performers in a periodic assessment of function prediction for protein families. So NetGo2 and DeepGo+. So these are different machine learners built to predict protein function in a similar way for individual organisms. Brighter colors are better. And so you can see, first of all, Fugassem has more colors. We can do this for any bug that is sufficiently prevalent in any microbial community. And second of all, there's a lot more red in there than we expected. And I thought at first, again, okay, this seems too good to be true. What's performing so well? Particularly when we combine data types in Fugassem, we started getting surprisingly accurate predictions. And this seems to boil down to 2 things. One is that combining information across multiple bugs in whole communities was surprisingly helpful. So if you see the same kind of homology patterns or domain patterns in several different bugs, that tells you a lot more than if you just see it in one isolate at a time. And second of all, the part that really made a difference is that combining data types matter. So if you have, say, 60% amino acid homology, that's not enough to know that 2 proteins do the same thing. If you see some co-expression, it's not enough to know that they do the same thing. But if you see both of those things at the same time, suddenly it's very synergistic. Suddenly that's a much stronger hint that, yeah, those 2 proteins are in the same pathway or working on the same substrate. So that turned out to provide a big performance boost for Fugassem. Another result that I thought was wrong at first is that in the process of now applying this to things like the human gut microbiome, green here are new annotations, either high confidence in dark green or medium confidence in lighter green. We can assign fairly large fractions of many different organisms' pangenomes to at least some putative function this way. But some of the best predicted organisms are those for which there are many isolates and kind of good isolate experimental data. And E. coli popped to the top, and I thought that can't be right. We know everything about E. coli already. We can't possibly be predicting that many new gene functions. And when we dug into it, the pangenome of E. coli in your gut or in 1,000 different people's guts is so large compared to the pangenome of isolates that have been well characterized that, yeah, we actually do transfer pretty well lab knowledge from the small number of lab strains out into the larger pangenome of wild E. coli pretty accurately because E. coli has been well studied. So even for these organisms where we know a lot about their isolates, and in some ways, especially for these organisms, we're able to transfer some of that knowledge out into new proteins with some sequence similarity and other functional data out in real microbial communities. And to keep zooming in here so you have a sense of, yep, it's not just performance numbers, we're actually seeing specific proteins doing specific things. We can go into your favorite organism and your favorite pathway and pick out proteins that are probably new members of that pathway. So I mentioned F-Praus earlier as a very stereotypical health-associated microbe, where if we're interested in things like its phage interactions, like we saw earlier with Totatus virus, each orange or red protein is a protein that's been newly placed into one of these either phage defense or CRISPR-specific systems in association with one or more proteins that anchor it into that pathway. So proteins where we do have expectation that that's what they're doing. The same thing is true for the bad guys. So Hungetella is a bug that I think I mentioned that tends to be enriched during colorectal cancer. Here, if we're interested in things like motility, which is often kind of disease phenotype or inflammation associated, or B vitamin biosynthesis, which is a very human microbiome characteristic process. Most different environments across the human body interact somehow with B vitamin biosynthesis. Here we can start placing individual proteins into these pathways as well. So we can zoom all the way from these very large high throughput whole community data sets down to individual proteins in individual pathways. And of course, very, very few of these we followed up with a small number experimentally. They do check out, that's good. This doesn't replace experimental follow-up, but it does give a leg up on those 1.6 million proteins, and we can at least start chipping away at them computationally to then accelerate and follow up on some of this huge amount of microbial functional dark matter. I'll finish up with a third different type of dark matter that's particularly fun for this audience, since we've been working with Hill's Pet Nutrition on a project called the One Health Microbiome Resource, which we unfortunately named before the center here launched. And Seth and others were very kind about kind of harmonizing the naming. Since this is a much smaller initiative than everyone has here, again, in collaboration with our research team at Hills. And one of many reasons we got interested in the companion animal microbiome is the degree to which Hills and the broader industry really care about companion animal nutrition and diet in a way that we can never do for humans. So we've been working mostly with data from the Hills Global Nutrition Research Center, which is this beautiful facility in Kansas, outside of Topeka. Diakar that we work with there refers to it as kitty and puppy heaven. There's about 1,000 dogs and several hundred cats whose only purpose in life is to eat different foods and see which ones they like. So it's a beautiful facility. We've been working with about a little more than 2,000 shotgun metagenomes that were specifically generated by Hills, plus another 500 or so that we've meta-analyzed from the literature, grouping these together into the One Health Microbiome Resource. So as kind of an aside, if you're interested in any of that data, metadata, assemblies, other information from companion animal cat and dog gut microbiomes, we've tried to provide all of this as a resource to the community. During the earlier talk, I spoke about some of the more basic biology or ecological analyses we've done with those data. In the last couple of minutes here, I'll focus on one of the more specific studies that really show off the kinds of nutritional information we can get from animals that people just won't tell you or can't tell you in that much detail. So one of the experiments that an additional collaborator, Matt Jackson, I forgot to put his picture on the slide, designed was a crossover feeding study of different types of low carbohydrate diets, specifically in dogs. So we had a collection of a couple dozen dogs. I didn't put that number on the slide either. That had a 1 week pre-feed on kind of a typical dog chow that's relatively high carbohydrate. And then were split and then swapped in one of two orders, either a low carb, high protein diet, followed by a low carb, high fat diet, or vice versa. And you can see that in the overall nutritional composition. I'll keep this coloration for the next few slides. So red is always high carb, purple is always high fat, and blue is always high protein. That's sort of the dominant energy source in each of the three different diets. And of course, I've cut off here, we know the rest of the macro and much of the micronutrient composition of these diets as well. Unlike some of the other dietary interventions we've done, so we did a similar intervention with different fiber sources, surprisingly small effect on the microbiome. This intervention had a gigantic remodeling effect on the microbiome. So again, all of the red dots are quite separate from all of the blue dots, are quite separate from all of the purple dots. Even at a very high level of which bugs were how abundant, we can find bugs that are dramatically more abundant specifically during the high fat diet, dramatically more abundant specifically during the high protein diet. This is on a log scale, so these are a couple log changes. Dramatically higher during the either of the low carbohydrate diets, or dramatically more abundant during the high carbohydrate diet. So out of the couple of different dietary interventions we've been able to look at so far, this had just a huge remodeling effect on the entire gut microbiome. But even underlying a big shock, a big impact like this, it turns out there's some surprisingly detailed ecology, there's some surprisingly detailed biochemistry. One of the things that we were surprised by is that there are a handful of bugs that show priority effects. So here's one of the most dramatic examples where for this particular uncharacterized firmicute species, it starts at a very low abundance across the entire population. So it doesn't really like whatever the pre-feed is, high carbohydrate diet. And if it sees the high fat diet first, followed by the high protein diet, doesn't care. Nothing happens. It doesn't really expand in the microbiome. If it sees the high protein diet first, followed by the high fat diet, it immediately becomes significantly more abundant, and in many cases stays more abundant even after the transition into the high fat food. This is the most dramatic example, but there are about a half dozen bugs that show these really kind of ecologically surprising priority effects, where the order in which they see nutrients is somehow locking in the overall ecological regulation of the community and allowing that bug to say bloom in one case when it can't in the other, even given the same nutrient substrates. And in terms of those nutrient substrates, probably unsurprisingly, we have fecal and serum metabolomics for this data set as well, and those were even more dramatically changed than the composition of the microbiome in the first place. So if we ordinate the fecal metabolomics, even more separation between the three diets, and there's still a lot of really surprising details going on under there. So a pet peeve of mine is that many microbiome papers will lump together short chain fatty acids and just say short chain fatty acids were up or down, or short chain fatty acid producers, as if there's one specific set of microbes that do that. If we have actually detailed targeted short chain fatty acid measurements here as well as untargeted metabolomics, butyrate, for example, was significantly increased in the high carb diet, which that you might expect. Acetate, and I'm not showing propionate, but acetate did basically nothing. Propionate actually tips a little bit in the opposite direction, and even very closely related fatty acids like isobutyrate are actually going in the opposite direction. So it's up more in the high protein diet. So it's often incorrect to lump together, say, all short chain fatty acids, all branch chain fatty acids, all bile acids. Microbes are always really focused on fairly specific substrate chemistry, and that's true even for these kind of classically bioactive molecules like the short chain fatty acids, and it's true even in a huge dietary perturbation like this. One of the other larger effects was on, I'm debating, yeah, I'll do this and then skip the next couple of slides for time. One of the other larger metabolomic effects was in amino acids, where here they do sort of behave as a group. All of the amino acids were generally depleted among both of the low carb foods, especially the high fat food. And these all correlated, aside from a couple of outliers like cysteine, were all quite correlated with each other. And this in turn corresponded with a variety of different bugs that tended to correlate with increased or decreased amino acid abundance across the board in both diets. So what you're looking at here on the x-axis is how much each significant bug correlated with amino acid changes during the low-carb, high-fat diet. And on the y-axis is the same thing for the low-carb, high-protein diet. Most of these are on the axis. So most bugs were amino acid correlated in both conditions if they were in at least one. And if I call out just this orange point here, this is again an uncharacterized Oscillospiraceae species, dramatically anti-correlated with amino acid levels. For anyone who's looked at this type of microchemical interaction, this is far, far stronger than what we see in, say, any human dietary study. So if you tend to have this bug, then your amino acids are dramatically reduced. If you don't have this bug, then residual amino acid levels in stool can go quite high. And that was true again, mostly in the low carb, high fat diet, but to a strong degree in the high protein diet as well. And despite having just recently submitted this paper, we're still really not entirely sure why. It's not clear why all of the amino acids would correlate, and it's not clear why they would come up even in the high fat diet, which does not have a particularly enriched protein content compared to the high carb diet. So we assume that it's something to do with lower residual carbohydrate substrates available. We kind of sort of thought maybe that microbes were broadly shifting from carbohydrate metabolism to protein catabolism. On one hand, this agrees with an enrichment for amino acid metabolizing pathways and protein catabolism pathways in the metagenomes. On the other hand, it's not supported by the residual nitrogen measurements in the stool. So it's not clear that we don't have appropriate levels of nitrogen in the stool metabolomics to support this being just a protein catabolism event. So we're not sure why this is one of the main microbial metabolic remodeling consequences of this particular dietary shift. So I'll end on that mystery. I'll skip the up next example that we have here. We're just starting a project that looks at puppy development, where we have both social contact information and very dense longitudinal time courses, along with these extremely detailed dietary records. So again, kind of just quick sneak preview for each dog. We know every day for the first several years of its life what it ate, how much it ate, what the nutrient composition is. We can classify those into different broad categories of food type. We know where those dogs were housed, what kind of outdoor exposure they got, what medications they got. So Hills has been just a tremendously helpful collaboration partner, both because they do great science and because we cannot get this information from people. So stay tuned. We're just starting that, along with some other interesting studies looking at antibiotic response. So can we improve the recovery from antibiotics with different dietary interventions, or looking at some of the dark matter, biochemical dark matter that's specific to the canine and feline gut microbiomes. A lot of this is work supported by our particular microbiome center at Harvard, which is the HCMPH, or Harvard Chan Microbiome and Public Health Center. We do, as I have learned, we do similar things to a lot of the activities here. One brief advertisement that tends to be a little distinct from other microbiome centers is that since we do so many population studies, we've built up both a physical sample collection platform and a sort of automated bioinformatics and biostatistics platform specifically for handling microbiome data. So we have a menu of different sample types and modalities we can collect. If you have a need to collect tens of thousands of stool samples at room temperature without needing overnight shipping for different molecular assays or for culture and notobiotics, that's a thing we can do. So feel free to follow up with the center directors. And another activity we have coming up is that we just recently finalized the date for next year's symposium, which Erica joined last year. So we don't have a registration up yet, but save the date for next May 18th. This is a hybrid format. So if you want to come to Boston and join us, great. If you want to watch online, keep an eye out for when we have the Zoom registration up. And if you do want to come to Boston in person for a hands-on workshop for microbial community bioinformatics, we're hosting our second local version of that next year. We just saved the date in June. And again, we don't have this year's registration up, but you're welcome to look at last year's curriculum and information on the center website. With that, again, I'll thank our collaborators. Anyone whose picture you saw on the slides on the right from Romnick, Wendy's group, the Hills team, Owen's team in Maryland, Nicola, and then of course Wendy and Long Nguyen was also involved in that cancer study. We've had the pleasure to collaborate with several different groups on these projects, and then everyone on the left whose pictures you saw were the folks from the lab who did all of the cool work. So if there's time, I'm happy to take questions. And again, really glad to join the group today. Thank you.
I think we have time for a couple questions, if anybody has any. First question typically comes from a trainee, so feel free to ask.
For the baklava tool that you presented early on in the talk, have you tried this tool in non-human systems, and kind of what has the success been for taxonomic idea viruses?
Good question. So if I heard right, it was have we run it in non-human systems and what happens?
Yeah.
Only barely. So we are in the process now of the study I mentioned from Kelsey and Will Nicholas should about as well. They just finished the compare everything else on different environments study. They're just running baklava on those samples. I know it's still mostly phage. We do not yet have an estimate of how much is unknown. So we don't know an estimate of what fraction of viruses we're not seeing in those environments. I expect it to be much higher.
Gotcha. Thank you.
I have a question about the puppy study. Are those all facility dogs, or are they being integrated into house?
They're facility dogs. One of the nice things about it is that we enroll them as they come into the facility. So I skipped over the slides, but they're enrolled as cohorts. So there's some correlation between age and cohort and housing, but we do capture them right as they come in, and then over time, they start to be more mixed. So the colors on the right here are their locations over time. And you can see each cohort starts in one of a couple of puppy facilities, and they're gradually transitioned into these beautifully structured rooms that they have there on a per room, per subpopulation, and per pin basis. So it's the level of location detail under that figure is, again, just amazing.
Do you sample any of the humans that interact with the dogs?
We're not. That's a very good idea. The techs there are also just amazing. They know the dogs by name. They are very good at collecting the canine samples. We don't have IRB to do anything with the people.
Thank you for the talk. Wonderful. I learned a ton as usual. So for following up on Erica's question, is there an end game to the Hill study about how they're going to use this information in products or care or personalized pet therapies, if that's coming someday?
Their motivating interest is very much to incorporate microbiome technology as their term into their products. We got involved because a couple of years ago now, they launched a microbiome targeted food it's basically fiber enriched. So it's a good example of where we don't really know the mechanism of action, but adding fiber, and it was specifically targeted at canine enteropathies. Adding fiber helps dogs have fewer stomach problems. That's kind of what it boils down to.
Yeah.
They're trying to get a handle both on things like mechanism of action. So what's specific about those fiber substrates or that combination is active in that case, to expand that in a more specific way. So like the developmental study is a good example. How can this help puppies around the time of weaning? The antibiotic study is targeted in a similar way. So can we, especially since there's so much veterinary antibiotic use, can we design foods that are better at supporting safe recovery after antibiotics? So they're all centered around, the fiber study was based on that original product. Like, can we narrow down or target the subset of fibers, either for that product or for different indications.
Yep.
So they're all based around that kind of product outcome eventually.
Nice. And I'll be curious to see if there's any thinking on whether staying on one diet for a long time versus alternating diets is leading to differential health outcomes that could inform those strategies. Because most of us just have our dogs on one diet for a very long time.
That's something that we've had to incorporate into the antibiotic study. And it's kind of a pain in that context. Some of the dogs coming into it have been on palatability tests, which means they shift quickly between diets before whatever happens. So these are all clinically recommended antibiotics. So if they get a scratch while they're playing or something like that, some of the dogs have been on different diets leading into it, and some of them have not.
Yeah.
So on one hand, it's a pain because it cuts down our effective sample size. On the other hand, we can test things like that.